Dr. Leandro Martinez. He works from the Institute of Chemistry in Campinas, Brazil, and he's going to talk about his work. So thank you for coming.
Okay, thank you very much, Carlos, for the invitation, and everyone at the University of Autónoma of Mexico. As we were talking offline that we could do this presentation in Spanish, so I encourage anyone to ask in Spanish questions afterwards if that is more comfortable. So, but we will present in English such that a wider audience can follow the talk. So I'll be talking about protein solvation in complex molecular systems. We work at the University of Campinas, as Carlos mentioned. We are part of the Center of Computing and Engineering and Sciences of the University of Campinas. And this is our group's homepage. Everything that will be described here is also available at this homepage. So what's the central problem that we are going to speak of is protein stability and its relation with the solvation of the protein. So there is a general view which correlates the environment in which a protein is present and the stability of this protein. And the general view is the following. If the protein is solvated in a mixture of solvents in which one of the solvents or the co-solvent, which might be alcohol or sugars or urea or denaturants or stabilizers, if the co-solvent interacts favorably with the surface of the protein, it tends to favor structures with greater surface area. Therefore, a co-solvent that interacts with the surface of the protein very strongly tends to favor structures with greater surface area, thus promoting the protein denaturation, which, because denaturation generally leads to greater surface, to the unfolding, and these unfolded structures have, in general, greater surface area than the folded structures. In these situations, we say that the protein is preferentially solvated by the solute, by the co-solute of the solution. For example, by urea. Urea is a type of co-solute that interacts strongly with the surface of the protein. On the other side, there are co-solvents that do not want to interact with the surface of the protein, or in other words, that the interaction of these co-solvents with the protein surface is not favorable. Therefore, the protein remains preferentially hydrated. So there is a depletion of this co-solvent from the region vicinal to the surface of the protein, and then since the protein the protein surface does not want to interact with the co-solvent, the presence of the co-solvent favors structures with a more globular form, with smaller surface areas. Therefore, there are co-solvents that, by avoiding the interaction with the protein surface, promote the stabilization of the folded structures, which in general have smaller surface areas. So this is the general picture. There are proteins in mixtures of solvents. The solvents or co-solvents that interact favorably with the protein surface tend to increase the protein surface, thus promoting denaturation, and the solvents that avoid the protein surface tend to promote the compactification of the structure, thus in general favoring the folded structures. We want to study this phenomenon from a molecular level. So in general, when we want to understand the structure of a solution from a molecular level, we compute distribution functions. The most common distribution function is the radial distribution function, which computes the density, the relative density of some species in the solution relative to the bulk solution. But these distribution functions, the radial distribution function, in general is computed from the center of mass of the solute. This is one possibility. And this is very bad in terms of visualizing the molecular structure of the solution, because for example, an irregularly shaped solute like this one will display solvent molecules in this region, which are relatively close to the center of mass of the solute, which is more or less here. And on the other side, there will be solvent molecules which are also on the surface of the solute, but are far from the center of mass of the solute. So if we compute the density of the solvent relative to the distance of the center of mass of the solute, if the solute is very, very irregularly shaped, this does not provide a clear picture of the interactions of the solvent with the surface. So we want to compute a property from the solutions, from the structure of the solution, that takes into account the fact that the solute and the solvent might be molecules with complex molecular shapes. So here we have been using and proposing the use of minimum distance distribution functions for this, with aiming this representation of the molecular interactions at a molecular level. So what do we compute? We compute for each solvent molecule, we compute the minimum distance between any solute atom and any solvent molecule and any solvent atom. Therefore, and we start to compute which is the density of these minimum distances as a function of the distance from the solute, the distance computed as the minimum distance between any solvent and any solvent and then any solute molecule. For instance, if we compute in this case, we have a solution, a protein, in this case, which is in a solution of water and TMAO. TMAO is a known protein stabilizer. And we compute the minimum distance between any atom of the TMAO molecule and any atom of the protein molecule. We will find that there are some of these distances which correspond to a hydrogen bonding distance, a hydrogen bond between this oxygen atom of the TMAO atom and some hydrogen bond donor of the protein. The minimum distance distribution function will clearly display this peak, which here is a shoulder, more or less, this peak, which corresponds to finding molecules of TMAO at a hydrogen bonding distance from the protein. And this is very good because this is a clear representation of the surface level or solvent shell level interactions between the solvent and the protein. And this is independent of the complexity of the solute and of the solvent shapes, of the shapes of the solute and the solvent, because we are computing the shortest distance between any atom of these two species. Also, TMAO displays a larger peak at intermediate 2.5 angstroms, more or less, which represent essentially, as we will see here, the interactions of the methyl groups of TMAO with the hydrophobic groups of the surface of the protein. This minimum distance can be decomposed, actually, in the contributions of each type of atom with the solute. So here we decompose the distribution function, which is the black line that was obtained for the complete TMAO molecule, in the contributions for this black curve of each type of atom of the TMAO molecule. And we see that the first peak is essentially composed by contributions of the oxygen atom, because this is the hydrogen bonding atom of the TMAO molecule. This makes sense. This makes sense from a molecular level perspective, from a chemical perspective of these interactions. At the same time, this secondary peak, which is stronger and represents less specific interactions, is essentially dependent on the distribution of the hydrogen atoms of the TMAO molecule. So we obtain both a molecular picture of the solvation and which is the orientation of the molecule on the surface in some sense, because we can extract from the distribution function, which is the black line, which are the groups of atoms or atoms that are contributing to each of these peaks in the solvation structure. For instance, one example of the application of these distribution functions to understand solvation, let's go for the case of urea. And in this case, urea is a non-denaturant of proteins. So the expectation is that urea will interact strongly with the surface of the protein, promoting denaturation, favoring structures with protein conformations with greater surface area. So this is what we obtain as the distribution minimum distance distribution function of urea relative to a protein. And we see clearly that there is a very strong peak with a great density augmentation of urea in the vicinities of the surface at about 2 angstroms from the surface of the protein, which is consistent with the fact that urea interacts favorably with the surface of the protein. Okay, this is expected, and urea promotes denaturation because these strong interactions intend to increase the surface of the structure, promoting denaturation. But now we compute the same distribution function, the same type of distribution function for TMAO. And TMAO is known to be a protectant, so TMAO is expected to stabilize the protein structure. However, if we think from a chemical perspective, we should expect that TMAO is able to form hydrogen bonds with the protein structure because it has a hydrogen bond acceptor atom. And also it has a more or less hydrophobic part. This molecule has a more or less a hydrophobic part, which we expect should interact also in some sense favorably with the more hydrophobic regions of the surface of the protein, which also may have this characteristic. Thus, when we compute the minimum distribution function of TMAO relative to the surface of the protein, what we obtain is something that qualitatively is not that dissimilar from what we obtain with urea. We do see that TMAO accumulates on the surface of the protein, forming specific interactions, both hydrogen bonds and non-specific interactions through the methyl atoms or methyl groups of TMAO. Thus, there are specific interactions of TMAO with the surface of the protein, which is in agreement with our perspective from a structural point of view. However, in some sense, this is contradictory with the fact that we expect that TMAO should protect the protein by promoting preferential hydration. We expect TMAO to be excluded from the surface of the protein in such a way that it will promote the stabilization of structures with a smaller surface area. In order to actually get the correct picture, we need to connect these distribution functions, which are a molecular view of the interactions at atomic level, with the distribution or the properties at a macroscopic level. So, and for that, we need to integrate those distribution functions in the sense that we need to measure effectively, given a region around the solute. For example, this is the protein. This is a region around the protein, which essentially encompasses everything in the solution that the protein affects by its presence. So what we are calling here the solute domain is the region of the solution in which the molecules that are present in this region are affected by the presence of the protein in this solution. So we know that both urea and TMAO interact with the surface of the protein, but this does not necessarily mean that urea will be in general with a greater density in this gray region, or the fact that TMAO can interact with the surface of the protein does not necessarily mean that TMAO will have a greater density in this gray region of the solution. Effectively, that is what we obtain if we compute the Kirkwood-Buff integrals. This is the technical name of this integral, the sum of the number of solvent molecules that are within the region that the protein affects in the solution. We sum the number of the molecules of the co-solvent in this region and check if the density of the molecules in this region is greater or smaller than the expected density for that region if there were no interactions with the protein. And the integrals have this shape, but what I want to highlight is that this is the integral for urea, and it says that, okay, urea is depleted from the region where the protein effectively occupies a volume because the protein has an effective volume. And then, because it has strong interactions with the surface of the protein, there is an accumulation of urea, which increases this integral. And at the end, what we obtain is that urea has a final Kirchhoff integral, which is greater than the corresponding Kirchhoff integral for water in the same system. And this is a theoretical development, well known for the people in the field. This means that urea solvates the protein more effectively than water in this system, because this integral is greater for urea than for water. In other words, from a macroscopic point of view, urea preferentially solvates the protein in this system, as expected. We expected that urea would preferentially solvate the protein, promoting denaturation. On the other side, TMAO is also excluded from the protein volume, and it accumulates on the protein surface. This rise of the integral is associated with the accumulation of TMAO on the protein surface. However, this accumulation is not enough for the density of TMAO in the region where the protein affects the solution to be greater than the density of water, of the associated density of water in the same region. This means that despite the fact that TMAO can form specific interactions with the protein surface, water is the preferential solvator of the protein in this case, or in other words, this protein is preferentially hydrated because the integral, the Kirchhoff integral of water is greater than the Kirchhoff integral of TMAO in this system. Thus, this is perfectly consistent with the macroscopic observation of urea being a denaturant because it preferentially solvates the protein, and TMAO being a protectant of the protein structure because it is overall excluded from the protein surface, leading to preferential hydration of the structure. This is interesting because thus we have preferential solvation of the protein, which for urea, which implying denaturation, in a situation in which there are, of course, specific interactions of urea with the surface of the protein. But at the same time, even if there are specific interactions of the protein with TMAO at a surface level, overall, the preferential solvation favors water, and the protein and TMAO is effectively excluded from the protein surface, avoiding the development of large surface area structures and leading to the compactness of the protein, which in general imply the stabilization of the folded structures. So this is the summary of the methods that we are using. So we have the systems consist of a protein or a complex macromolecule in a mixture of solvents. These solvents and co-solvents can promote the protein stabilization or protein denaturation. We use these minimum distance distribution functions to obtain a molecular picture of the interactions at a surface level, which is essentially independent of the structures or shapes of both the solid and solvent molecules. So even if the solvent molecules are very complex, even if the solid molecules are very complicated, we can, with the same calculation, obtain a clear molecular picture of these interactions at a molecular level. And we can compute preferential interaction parameters, these ones which are connected with experimental observables, and that have to be correlated with the macroscopic view of solvation, which says that the co-solvents are either accumulating on the protein region promoting denaturation in general, or being depleted from the protein region, promoting stabilization of more compact structures. So this is the overall picture of the methods we are using. Now I will describe some examples of these applications that our students have been developing in the last few years. The first example, as I was discussing, came from a discussion with Nina Pastor, there from Mexico, and it is the problem of trifluoroethanol stabilization of alpha helices. Trifluoroethanol is this small molecule which has a hydroxyl group which is able to form hydrogen bonds and trifluorines. And it's a well-known thing that this molecule promotes in general the stabilization of helical structures of proteins. We have studied this peptide, which is a trimer of this alanine-alanine, glutamine-alanine-alanine mer peptide. So it's a 15 residue peptide, which I took a nice picture of the folding and unfolding of this peptide from this article. And so we can see that there are helical structures, that are structures in which the helix is only partially formed, and there are structures where the helix is completely unfolded. And okay, it is a well-known fact, and this is a result from our simulations. I will describe this more clearly later, but essentially it's a well-known fact that if you put this peptide, which is a model helical peptide, in solutions of trifluoroethanol will promote the stabilization of the helix. So the blue line here is the peptide in water, and here we have the alpha helical content. And we see that, okay, in water, there is some profile of alpha helical content as a function of the residue. And in essentially any addition of trifluoroethanol, the alpha helical content increases, and thus effectively providing the picture of TFE stabilization of alpha helices. We found, but when reading about this problem, we found in the literature that there is a discussion on which is the molecular basis for this stabilization, and in particular, one mechanism called our attention, and which is a direct mechanism of TFE stabilization of the protein structure. And essentially, what this mechanism proposes is the following. The helix is stabilized by these intrahelical hydrogen bonds, which are formed between the amide carbonyl and the amide-amine groups. So this intrahelical hydrogen bond of the helix is fundamental for the stabilization of the helical structure. Water on this side can form hydrogen bonds both with the carbonyl groups, and it is small enough such that it can form also hydrogen bonds with the amine groups in such a way that it can intercalate in between these groups, leading to the breaking of this hydrogen bond, which is the critical hydrogen bond that stabilizes the helices. So this is the mechanism by which water can lead to the denaturation of a helical structure by intercalating in these hydrogen bonds. There was a proposal that the TFE could protect the alpha helices by forming one hydrogen bond with the oxygen atom of the backbone of the helix. But since the other part of the molecule is not a good hydrogen bond donor or acceptor, by forming this hydrogen bond, the presence of TFE here would be impairing the approach of water, and in such a way that water wouldn't be able anymore to as easily interact with this hydrogen bond, promoting the breakage and thus the unfolding of the helix. So we decided to probe, to investigate if this mechanism can actually be supported by data. So we wanted to check if this specific direct mechanism of helical protection by interactions of the trifluoroethanol molecule with the backbone, with forming hydrogen bonds with the backbone, could in fact effectively be the mechanism by which, or the preferential mechanism, the main mechanism by which the TFE molecules sustain the helical structure. So what we did is to perform exhaustive simulations with using a technique which is called Hamiltonian replicate exchange molecular dynamic simulations. What is important here is that we were able to obtain essentially converged ensembles of the helix coil equilibrium. So our simulations are long enough to probe the denaturation of the helix, the formation of the helix, the denaturation of the helix, the formation of the helix several times. So we have a complete sampling of the structures in the solution. And then we went on analyzing how these peptides interacts with the solvent here being a mixture of water and trifluoroethanol, studying the structure of the thermodynamics of these solute-solvent interactions. And then what we thought is that we could perform alchemical perturbations in the interactions of the system in such a way to probe specifically the role of that hydrogen bond in the stabilization mechanism. So what we obtain in simulations of this process of solvation of this peptide in mixtures of water and TFE is that, of course, water can form hydrogen bonds. This is a strong peak here in the distribution functions, is an indicative of hydrogen bonds of water with the peptide. This is expected, particularly the hydrogen bonds of water with the backbone are expected and are known to promote the denaturation of the peptide. TFE also forms strong hydrogen bonds with the peptide, and we will see where these hydrogen bonds are being formed. And this is consistent with that picture that TFE might be stabilizing the helical structures by interacting with the backbone of the peptide. So we do have hydrogen bonds between TFE and the peptide. And in additionally, when we compute these Kirkwood-Buff integrals, we see that the Kirkwood-Buff integrals of TFE are greater than those of water, which means that the peptide is effectively solvated by TFE preferentially. That means that this co-solvent, trifluoroethanol, preferentially binds the peptide. Let's remember that in general, co-solvents that preferentially bind the structure tend to unfold the structure. Here, we don't have this situation. TFE is a helix stabilizer that's clear and that appears in the simulations very clearly, but TFE is a strong binder to the surface of the peptide. Thus, certainly this is not the case of a typical mechanism of protein stabilization. And it also seems to suggest that the specific interactions of TFE with the protein structure should be responsible for this stabilization. And this is all consistent with that picture that TFE might be binding the backbone, and by binding the backbone of the protein, impairing the approach, hardening or hampering the approach of water molecules to the surface of the structure. Okay, so this seems the picture is quite consistent. So here we compute the minimum distance distribution function of TFE relative to the peptide, and there are hydrogen bonds. These are this peak corresponds to hydrogen bonds. These hydrogen bonds occur through the hydroxyl hydrogen of TFE, which is this one. So perfect. The hydrogen bonds are occurring, of course, by means of this hydrogen atom. And we see, and if we look at the contributions of the peptide, of the backbone of the peptide to the minimum distance distribution function, we see that the backbone of the peptide is forming hydrogen bonds with TFE through the carbonyl oxygen, which is in perfect agreement with this picture of TFE forming a strong hydrogen bond with the carbonyl of the helix, and being in certain sense stable at its position, which was interpreted as the reason why water cannot penetrate here and stabilize the helix. So what we thought to do, okay, let's confirm that this interaction is the interaction that is causing stabilization. So what we did is to modify the force field of the simulation in such a way that this hydrogen bond, specifically this hydrogen bond, could not be formed anymore. So we increased the van der Waals radius of the interaction associated with this oxygen and these oxygens of the helix in such a way that this specific interaction could not be formed anymore. Our modifications of the force field only affected this specific possibility of forming a hydrogen bond. So essentially, we performed simulations again in which this hydrogen bond was not possible anymore. And what happened is that water continues to form strong hydrogen bonds with the structure. Actually, if we look at the picture that was in the previous slide, we see that water interacts even strongly with the peptide, which is okay, is expected. We see that, okay, the hydrogen bonds of TFE with the peptide almost disappear of this hydrogen bond. There are still some hydrogen bonds of the peptide of TFE with the side chain of the glutamine residue, which are the remaining hydrogen bonds here. So we effectively eliminated the hydrogen bond between TFE and the backbone of the peptide, and we expected to see that the helix will be destabilized. And what we have seen is that completely the opposite effect. By removing the hydrogen bonds between TFE and the backbone of the protein, the stabilization effect of TFE was much greater than when this hydrogen bond is present. Therefore, what these simulations show is that these interactions between TFE and the backbone of the protein are destabilizing interactions, as are the hydrogen bonds between water and the backbone of the protein, also destabilizing interaction. So every competing hydrogen bond between the intrahelical hydrogen bonds and the solvent will destabilize the helix. In this case, TFE was effectively contributing to helix destabilization by forming this hydrogen bond, despite the fact that, as an overall effect, TFE is stabilizing the helix. What do we have in practice is that, okay, TFE forms more interactions, particularly non-specific interactions, those interactions that are not hydrogen bonds, with the helical structures than with the non-helical structures. So TFE likes to solvate the helical structure. So it interacts favorably with the helical structure more than with the non-helical structures. And this occurs most importantly in the non-specific interactions, not in the hydrogen bond interactions of TFE with the backbone. So TFE essentially solvates the hydrophobic character of the protein, of the helix, and by solvating this helix and excluding water, but not specifically excluding water from the hydrogen bonds with the protein, but excluding water in general from the vicinities of the peptide, is that TFE stabilizes the helix. If TFE was able to stay around the protein, avoiding water, without forming the hydrogen bond with the peptide, it would be an even greater stabilizer of the helix of the helical structure. I'll be more brief in this other example, which is an interesting application. Also, we have studied quite extensively the role of ionic liquid ions in solutions where there is a protein present. And in this particular case, we have studied the role of different ions that are used to compose ionic liquids, different cations, different anions, in the solvation structures of protein folding states, but in protein folding states varying from the folded protein to partially denatured protein to a completely denatured protein and to an extreme of a completely extended protein state, in which represented the maximum possible surface area that the protein peptide could have. What is interesting here is that these minimum distance distribution functions can be used to study the complexity of these solvation structures independently of their actual conformation. So we can use exactly the same technique and even compare, for example, the fact that the native structure here in blue forms relatively more specific interactions, hydrogen bonds, than the denatured states. Relatively, and the non-specific interactions are more prominent in the denatured states than in the native states, that is the blue curve, which is sort of expected because as the protein denaturates, the more hydrophobic residues are exposed, and thus we expect that the more hydrophobic interactions start to predominate relative to the more specific hydrophilic interactions that appear in the hydrogen bonding region. So minimum distance distribution functions allow the analysis of these details of the solvation, but also to compute the preferential interaction parameters that what's effectively connected with the macroscopic effect of denaturation or stabilization. So here we have an example. We have the preferential interaction parameter of one of those ionic liquids with the protein. And we would just say here that, okay, if this preferential parameter is positive, it means that the ionic liquid is preferentially solvating the protein, which means, as a general rule, that these interactions are favorable in such a way to favor structures with a greater surface area, thus promoting denaturation. When this is the orange rectangle here, when the preferential interaction is negative, we say that the ionic liquid is being excluded from the protein surface, thus favoring more compact structures, in general, meaning stabilization of the folded state. So, when the protein, if we look at the profile of the preferential solvation parameter for the native state, what we could infer is that the ionic liquid denatures the protein, is a denaturant co-solvent for smaller concentrations, but becomes a protectant co-solvent at larger concentrations. This is the picture that would be obtained from a typical analysis of solvation of a native state of a protein and the general picture of the role of co-solvents in protein stability or denaturation. However, we can actually compute the effect of the co-solvent, the variation of the concentration of the co-solvent on the equilibrium constant of the denaturation process. So if we imagine that we have a two-state mechanism in which we have the folded protein and the unfolded protein, this mechanism has an equilibrium constant, and we are computing here what's the effect of the concentration of the co-solvent on this equilibrium constant. And in fact, this effect on the equilibrium constant is just the difference between the preferential interaction parameter in the final state of the transformation minus the preferential interaction parameter in the initial state of the transformation. So thus, to actually understand if a co-solvent has a protectant or denaturant effect, we need to compute the difference in the solvation properties of the co-solvent in the denatured state versus the solvation properties of the co-solvent in the native state. And if we compute that, Even if we have regions where the native state is preferentially solvated by water, the ionic liquid is preferentially excluded from the protein surface, even if we have that for the native state, since the denatured states of the protein are so much more strongly solvated by the ionic liquid, this difference is always positive, and the ionic liquid is always a co-solvent that promotes denaturation from a thermodynamic point of view. We can still have a kinetic effect of protection, because at the beginning of the process of denaturation, the ionic liquid in higher concentrations want to be excluded from the increase of the protein surface, because the protein surface of the native state is not that friendly for the ionic liquid. But as the protein starts to denaturate, the exposure of the more hydrophobic core of the protein leads to the complete denaturation of the structure very favorably, because the ionic liquid interacts very strongly with the protein structure in the denatured states. I won't go into detail, but one interesting thing is that from these simulations, we can compute the contribution of any chemical group, in particular each amino acid residue, to this solvation structure. So we can obtain a very detailed picture, or which are the regions of the protein that interact more strongly with one or other ion involved in the interactions in the solution. So we can compute out this decomposition of interactions and obtain a very, very detailed picture of the solvation structures of the solution. How do we compute all that? So we are computing minimum distance distribution functions, and then preferential solvation, Kirkwood-Buff integrals, preferential solvation parameters. We have developed a software that does all that for us, which is called ComplexMixtures.jl. It's available for free at GitHub. This is the site. Essentially, this package is very simple to use. I hope so, at least. So this is a the Julia programming language package. So it's a free language, so you can just download it in any computer of any type and load the package, install the package, load the package, and use a typical input file like this one. Here we just load the PDB file of the system, then we define what's the solute and what's the solvent in the system, and here we load the trajectory. The package is compatible with the most common trajectories. Here I'm loading an AMD generated trajectory, but we can do the same for Gromax trajectories and so on, Amber trajectories. And here, with this command, we obtain the results. The results here are the combination of everything I have said so far. So this result structure will contain the distribution functions, the decomposition into atomic contributions, the Kirkwood-Buff integrals, and so on. And so this package allows obtaining all these pictures that I have presented present so far. Just to illustrate the potentiality of the package with very, very briefly some other examples, we have, for instance, one example where I have a polymer. This is polyacrylamide solvated by dimethylformamide. And what we see here is that even if we have a polymer which is essentially an unstructured macromolecule, we can obtain a clear picture of the solid-solvent interactions. We can decompose the interactions in each type of group that compose the solute, in each type of group that composes the solvent, and so far, and also in each group of the polymers. So here, for instance, we see that which is the density profile of the solvent, which is dimethylformamide, around each of the mers of the polymer, and in particularly we see that the central mer is less solvated than the mers at the extremities of the polymer, because the polymer has some level of folding which protects the center of the polymer from the solvent. Another example here is a different type of application. We have a membrane, lipid bilayer, which is here solvated by a mixture of water and ethanol. Ethanol is known to be a destabilizer of these bilayers. And we can, again, using the same ideas, we can compute the minimum distance distribution functions of the bilayer relative to these solvents. And you can see, for instance, that this would be the purple line here, that ethanol interacts quite strongly with the apolar chains, with the aliphatic chains of the surfactant molecules, while water does not interact at all with the surfactant chains of the surfactant molecules. So what we see is that ethanol is actually penetrating in the membrane and interacting deeply into the membrane with every chemical group of the surfactants. This can be seen also from the surfactant point of view. So here we have olive oil and palmito oil, and this is the density of ethanol as a function of the chemical group of each of the aliphatic chains. So here we see that ethanol penetrates quite deep into the membrane. It has some specific interactions which might be interesting here with the insaturation. The same thing for palmito oil. There's a deep interaction of ethanol in the membrane, while water is completely excluded from the interior of the membrane. So with the same strategies, we can obtain very detailed molecular pictures of interactions of these molecules in a complex system like a membrane with ethanol and a mixture of co-solvents and so on. Okay, this is an overall picture of this kind of study that we are performing. I must thank the founders of this work, in particular the São Paulo Foundation for Research, which finances the Center of Computing and Engineering and Science of UNICAMP, in which we belong. And you can access this site, which is quite interesting because it displays everything that the center is doing, and we have opportunities for fellowships and so on. This is our group. This picture is incomplete and outdated. There are other people in the group, and some people have left, but everything that I described here was developed by Vinicius, which is this guy, and by Ander, which is this guy. They are the ones that are working more extensively now with these solvation structures. This is Felipe, Pamela, and Guilherme. And okay, these are references. You can join our site to see the full text of the publications. And this is the site of Complex Mixtures. I hope I was clear. And again, if anyone wants to ask questions, I would be glad to answer, even if in Spanish, that's fine as well. Thank you very much.
Thank you, Leandro. So I'm going to start with the questions. I'm